Lyme disease has existed on the planet, has existed in North America for thousands of years. It was identified and named for the town of Lyme in 1977. When you think about primary prevention of any kind of a disease, you want to reduce the risk of exposure, maybe not be around ticks. And another important method of primary prevention is development of a vaccine. They started studying the bacterium that causes Lyme disease. There was a protein on the outer surface of the bacterium, OSP-A, outer surface protein A. Our immune system reacts to this OSP-A protein, and this became the focus for the development of a vaccine. Our immune system has a great memory, and when we are given a low, non-infective dose of a virus or a bacteria, our immune systems remember this. And the next time we're exposed to that same bacteria or virus, our immune system responds with antibodies. That's how most vaccines work. The Lyme vaccine works differently. The Lyme vaccine works inside the tick that's feeding on your blood before that bacterium ever gets inside your body.
The Lyme disease vaccine was based on an outer surface protein A of the spirochete. What we've learned is that spirochetes only express OSP-A in the midgut of ticks, and as they move from the midgut to the salivary gland during tick feeding, they no longer express this protein. A vaccine against Lyme disease that's based on this protein then requires that antibodies from the vaccinated host enter the tick midgut during tick feeding and kill the spirochetes in the midgut before the spirochete has a chance to move to the salivary gland.
The Lyme vaccine trials began in 1994. There were two major companies that had their own Lyme vaccine. Each recruited over 10,000 people to participate, half of whom got placebo and half of whom got the Lyme vaccine. So only one company actually went forth and got approval for to market the Lyme vaccine, and that was SmithKline Glaxo with Lymerix. And that came out on the market in 1998 and then was withdrawn in 2002. The reason being, cited by the manufacturer, was that there just wasn't enough interest.
People weren't buying it, people weren't using it, the sales were poor. However, it maintains FDA approval to this day.
For me, it was a very exciting project. The OSP-A vaccine was actually created at Yale, so it's a Yale story, and then went on to go from bench to bedside, so to speak.
The Lyme disease vaccine was important for two reasons. First, it taught us to actually look more closely at the biology of spirochetes and how they host adapt to their various microenvironments. Second, it also showed us that we have to be very careful when we develop vaccines in general so that we make sure we alleviate any public concern regarding safety of vaccines before they're mass marketed.
In addition to looking at ways to combat Lyme disease through vaccination, researchers are also trying to look at ways of controlling ticks.
Because this is a vector-borne disease, attacking the vector is really the primary mode of decreasing transmission. Researchers in the United States and in Europe are working on anti-tick vaccines that will either make you more aware of the tick bite or inhibit the ability of a tick to feed.
Some people can be seriously ill from Lyme disease, but the majority are treated very early on and do quite well.
The Lyme vaccine story is an incredibly complicated one, and prevention of Lyme disease continues to puzzle us.